Okay, thank you, Michael. It's great to be here, great to be talking about this stuff, even if it's online. So this is what I'm going to be talking about today, and I'd like to point out that it's actually Charlie Davis has done all the analysis on this long-term data set, so it's her glory, really. Generally in our group, what we've been trying to understand for quite a while is how different mechanisms of balancing selection act and interact together to kind of maintain genetic variation, especially in small populations, because obviously that genetic variation is very important. And that has brought us to looking at pathogen-mediated balancing selection, because obviously pathogens are such a fitness selection factor on hosts, and therefore that brings us to the immune genes that we're all talking about today. Which are great candidate genes to look at these processes with. And I'm going to be talking about work that we've been doing on the Seychelles warbler and the data set we've put together over about 25 years now, focusing on this small island population. The last population of the Seychelles warbler, there's about 350 birds that live on this island, and we've, for over 25 years, we've been tracking every individual on that island, monitoring their survival, their reproductive success, and obviously getting blood samples, etc., to look at their genetics. Now, in the Seychelles warbler, we've already done quite a bit of work. We know that this bottleneck population has low genomic variation overall, but we've also got clear signatures of variation being maintained at key immunogenetic loci. We've got strong signatures of selection at these loci. And this goes back to work I did on the MHC, including with Helena many years ago now. So we've got signatures there, but also on the TLRs, we've got signatures of selection. But I guess when we've detected these signatures and we've looked at their evolution over time, and found key evidence of balancing selection there, we also want to then know, are these signatures kind of ghosts of selection past, or is selection still acting on these key genes within this small bottlenecked population? And for the sake of today, we're focusing on one TLR3. Adrian will tell me off maybe for calling it viral sensing, but at least it picks up double-stranded RNA. We have a key SNP there in the LRR region of this TLR, and that SNP is at relatively high minor allele frequency. So for these reasons, we decided to focus and have a look at this TLR variant. And our aims are to characterize the evolution at this locus, to maybe determine what's driving that evolution, and in the long term, investigate why that variation has been maintained in this bottleneck population and what mechanisms are acting. We won't go into detail on the methods, but like I said, we've been catching individuals for a long time. And here we've got a subset of individuals that we've screened over 25 years to look at this locus and how it's changed over time. We use a basic SNP mechanism, calling mechanism, and these are the genotypes that we get. We have AC SNP, so we have AA individuals, AC individuals, and CC individuals in this frequency here. And the first thing we did was look to see whether we still had evidence that we're getting a change in those TLR3 allele frequencies over time. So we mapped it in adult birds over this 25-year period, and we got this consistent pattern of a decrease in the C allele over time. It's consistently reduced in frequency from 39 in 1993 to 28% now. Interestingly, because we've moved individuals from this initial source population to other islands as part of its conservation, we've got another 4 islands, and we can see from when they're established what happens to the C allele in those islands as well. And so we can see that we have this same general pattern of the C allele decreasing in frequency over time. Some islands it's faster than others, but you get the same general pattern. So we've got this contemporary evolution at the TLR3. We need to figure out what's driving it. So obviously the first thing we look at is whether the different genotypes are associated with differential survival. We have a clear picture here with the CC genotype having reduced survival, and that's in both sexes. There's no difference between the sexes. Leads to a significant decrease in survival, and a lot of that decrease happens in the first year or so. So then from that, we went on to see what impact that would have on the lifetime reproductive success of different individuals. We're screening all individuals, including, within an extra pair offspring. So we have a very good, accurate measures of lifetime reproductive success, because these birds can't leave the island, nor can their offspring. So what did we find? Well, for females, we find there's no significant difference in this data set between the different genotypes and lifetime reproductive success. But for males, we get a very significant effect, a very significant reduction in the reproductive success of the AC and the CC genotypes. So we've got TLR3 based selection happening, and it's happening especially on males, and that alone made us think that maybe there's other factors also occurring here, because we have this greater impact of TLR3 variation, but we know there's no differential survival linked to the genotypes between the sexes. So we thought maybe some sexual selection factors are occurring. We also find one other thing that was perplexing us, and that's we have quite a significant heterozygote deficiency in offspring in this population. So many less AC offspring than we would expect. So we then looked to see whether there was any selection, perhaps at the social level, whether males and females are more likely to associate with a similar genotype or less likely. What we did here was we worked out a distribution based on simulated pairs of what we would expect by chance in the population. And then we mapped onto that what we actually see in pairs. And so what you can see here is we have a kind of situation with assortative mating where individuals end up socially paired with other individuals which have the same genotype as them. And this is largely driven by AA females and AA males associating. We don't know yet who makes that choice or anything like that, but we're looking into that. We also looked to see whether there'd be any genetic mate choice, i.e. were females preferring perhaps to engage in extra pair fertilizations with males with different genotypes. But actually we found no significant difference either in extra pair paternity gained or extra pair paternity lost linked to the male TLR3 genotype. So the last thing we looked at was whether or not there was a link with post-copulatory selection. And here we just looked at the genetic parents that we'd already assigned. And what we did was look at situations where one parent was homozygous and another parent was heterozygous, so that we could see which allele was inherited from the heterozygous parent in these cases. Now, when we did that, first of all, we looked at heterozygote mothers and homozygote fathers. In that case, we saw no bias in which allele was being used or inherited from the mother. However, if we looked at a heterozygous father and a homozygous mother, we do find a significant difference. And we find that the C allele is less likely to be inherited from the father than the A allele. And this is obviously quite an exciting result, and we're screening many more individuals to try to follow this up now. So conclusions, well, we've got this contemporary TLR3 evolution, which backs up the signatures of selection that we've seen at this TLR3. We know it's partly been driven by natural selection, but we also know that sexual selection is playing a role both in pre-copulatory mechanisms and post-copulatory mechanisms. So that's our general conclusions at the moment. As you might realize, we haven't really got onto the mechanisms of balancing selection yet and unraveling them. But what we do know is there's no evidence of heterozygote advantage happening in this system. But what about the other mechanisms, fluctuating selection, rare allele advantage, etc.? Well, at the moment, we see this very consistent pattern. We see no fluctuations in this pattern at the moment, but we are only looking at a relatively short, well, extremely short period of evolution, obviously. We're going to try to extend that partly by going back to museum samples, which we've got about 26 of from about 200 years ago, and partly hoping that I live long enough and have a long enough career that I can keep sampling these things for many more years and see what happens. I think we forecast that the C allele at the present rate would go extinct in about 50 years. So I'm not betting on that one. Someone else will have to do that. But also we can look across the other different populations and see if we get different patterns over time. Yeah, so that's it. I'd just like to acknowledge everyone who helped, all the team that work on the Seychelles warbler, but mainly Charlie Davis, who successfully completed a PhD just a couple of months ago on this. So I'd like to say thank you and ask for any questions.